So this is a relaxed sarcomere we're going to zoom in and look at. These are the units of contraction in muscle, and this is how we get that striation. And this is showing you a relaxed sarcomere, and this is showing you a contracted sarcomere. And so what you can see, these things right here are called Z-discs. That's what's anchoring our sarcomeres. And in a relaxed muscle, look, here our Z-discs are out here. I can see this nice light band. I can see this nice dark band. In a contracted muscle, I pull those Z-discs together. My light band almost disappears. I'm all dark now. So I see that I've shortened. So here I was long, here I'm short. Well, these things here are all called myofilaments. And so we have a sliding filament model. So how do these filaments slide past each other? That's what we're going to be talking about. So in order to get there, we need to look at the anatomy of a muscle fiber. So we just talked about how muscles are arranged. Well, now then in each of these individual muscles, what is one of these muscle fibers made of? And if we look inside the muscle fiber, it's got even smaller structures, myofibrils. And if we look in a myofibril, it's got even smaller structures, myofilaments. Ugh. So that's what we're going to go through today, get through today. So, Sarcolemma. So as far as skeletal muscle fiber, we can, if we're thinking of something to call this is like fiber anatomy. And if we look at a muscle fiber, its plasma membrane is the sarcolemma. Okay, so that's one thing about it. And sarcoplasm is the cytoplasm. So if I look in here, this is my muscle fiber, I've got sarcoplasm. This is my cytoplasm, which means it's holding all of my organelles that are in my muscle fiber. Okay, some weird modified organelles that I find in a muscle fiber. These things called myofibrils. Okay, other things, modified organelles. In all of our cells, we have endoplasmic reticulum. In muscle cells, it's in our sarcoplasm, so I'm going to call it sarcoplasmic reticulum. And it's got these it's a really big branching network, and it's got these widened parts here next to these blue lines I'm drawing. So we'll talk more about that in a little bit. So this is sarcoplasmic reticulum, which I will from now on abbreviate as SR, Sarcoplasmic Reticulum. And this is modified endoplasmic reticulum that stores high levels of intracellular calcium. Anytime calcium's outside in cytoplasm, it's doing stuff. So in order to have high amounts of it inside the cell, not doing stuff, I've got to store it. So I store it in my SR. So we'll say this stores high levels of intracellular calcium. And my last modified organelle is this thing called a T-tubule, or a transverse tubule. And what it is this extension of the plasma membrane that goes down in and surrounds my myofibrils. So a transverse or a T-tubule are extensions of sarcolemma poking into and around our myofibrils. So my myofibril, we'll say for an organelle, contains my contractile filaments or myofilaments. Okay, so here's my sarcolemma surrounding my entire muscle fiber. I've got a lot of these myofibrils in here. This is, look, I can see my striations. So in there must be the basis for that. And then another thing we can see here, multiple nuclei. There are a lot of mitochondria also in skeletal muscles. So just be aware of that. Okay, so now our myofibrils. Myofibrils then are these cylindrical structures that we find inside our muscle fibers. And if we look at the features of our myofibrils, they've got these striations, which is this characteristic banding of light and dark. So if this is one myofibril, then what I would see is that I'd have dark and light and dark and light and dark and light and dark and light. So it would be striated. So the striations are just the banding of light and dark, and they're created by these things called sarcomeres. So that's what I want to draw now and talk about now, because this is going to form the molecular basis for muscle contraction. Okay, so everybody kind of know where we're at anatomically. We're zooming in, and we're getting smaller and smaller. Okay, that was a minute ago we had a whole muscle. This is one muscle fiber. It's got a bunch of myofibrils. So now we're going to zoom into a myofibril. They're awesome.
They are awesome.
Okay. So, the striations are caused by this banding of light and dark, and that happens because of these things called sarcomeres. So, sarcomeres. Sarcomere. What are the things that I need to know about a sarcomere? Well, if I'm just defining it, people say, oh, this is the span that goes from one Z disc to one Z disc. So here's a Z disc, and here's a Z disc. Another thing that I could say about a sarcomere is this is the smallest unit capable of contraction. So this is my unit of contraction. And it's composed or made of myofilaments. Thick and thin filaments. And it's the overlapping of these thick and thin filaments that gives us our striations, and it's their interaction that's going to shorten this sarcomere and cause contraction. So, for myofilaments, I've got two types. I've got thin and thick filaments. And so let's just look at this so we can get our striation and our sarcomere really quick, and then we'll zoom in. So, in the middle of my sarcomere, I have this region right here that's anchoring all of my thick filaments. We call this the M line. And if I look out from the M line, I have all of my thick filaments are kind of poking out from it. So for functions of the M line, or if I wanted to describe what the M line is doing, I could say these are proteins, or I want to say it's proteins. Proteins, they're perpendicular to the axis of the muscle, and they are anchoring my thick filaments. Well, if I look at the molecular composition of my myofilaments, then it'll tell me how these things interact. So, myofilaments. My thick ones are these ones right here. They're anchored to this M line. And if we zoom in and look at what these thick filaments are made of, they're made of these proteins called myosin. And myosin is interesting. These myosin proteins have these tails that curve around, and then they've got this double, I've heard it described as like a double-headed golf club. There are two heads on the end of each myosin protein. So thick filaments are made of myosin proteins. And if we were to zoom in here, people just draw pictures like this, but what I would see is all of these tails are running through here, and then I've got heads. poking out in 360 degrees. All around here, I've got these myosin heads standing up. So one protein just has its tail and its two myosin heads. But when I get a thick filament together, I'm gathering all of these proteins together, and in 360 degrees shooting out from that, I've got heads. All of these tails are meeting up in here. So I would just have a bunch of tails running in here, and all of my heads are poking out here. The head of a myosin protein has an actin binding site. Well, what's actin? It's the other protein that's involved in contraction. It's part of our other myofilament. So, yeah, let's just talk about it. So our actin filaments are thin filaments. Our thin filaments are our actin filaments, and they are all anchored to the Z disc and point in toward the M line. So this almost worked out as disastrous. So my thin filaments. Thin. They contain actin. They are anchored to the Z disc. And if we zoom in to a thin filament, actin is the protein that's involved in contraction. But we have to be able to make sure contraction isn't happening sometimes. So when we're relaxed, we've got these regulatory proteins that lay over actin and block the myosin head binding site. So troponin and tropomyosin are the two proteins that are involved. And this tropomyosin is this long protein that blocks all of our myosin head binding sites when a muscle's relaxed. Here's the myosin head. It's got actin binding sites. Here's the actin. It's got myosin head binding sites. Guess what those two want to do? They want to bond, and they want to bind to each other, and they want to start contracting muscles. But when I'm relaxed, I don't want that to happen. So this troponin tropomyosin complex blocks all of the myosin head binding sites on actin in a relaxed muscle. Troponin is just a protein that's involved in that. Calcium is going to bind it. And when calcium binds it, it moves. And when it moves, now I can contract. That's what we're all going to get to tomorrow. Contraction is really confusing and whatever, so we'll go through the nitty-gritty details tomorrow. Today I want you to understand the anatomy that's going to give rise to that physiology. We said calcium has to be stored intracellularly so that it doesn't do stuff. Well, when it's out, it's going to move that troponin or tropomyosin so that we can contract. So as far as our other things that I will say before I move away from this, is that our thin filament contains the regulatory proteins, or we'll say contains a regulatory troponin tropomyosin complex. So it contains the regulatory troponin tropomyosin complex. Okay. So now what I can see when I look at this picture is some different things that appear. This right here, this spot that's free of actin is called the H zone. This spot, where I have overlapping of my thick and thin filaments, is called my A band. This is dark. So when I'm looking at my striations, I have dark and light bands. My A band is my dark band. And I get that from right here, where my thick and thin filaments are overlapping. So myofibril features, striations, sarcomeres. The striations we can see, dark and light, and dark and light, and dark and light. All of that is coming because of this. So my dark, and if you're ever taking the MCAT, they ask you questions like this. In a contracted muscle, what disappears? Well, I'm going to lose most of my I band, which is light. I'm going to lose my H zone because I'm going to have overlapping of my thick and thin. So that's why I'm talking about it and why we need to know it. So A band. How you can remember that the A band is dark, or our dark band, is because there's an A in the word dark, right? So my A band is my dark band for striping. And that's where I have overlap of my thick and thin filaments. An I band is light. It also is handy that the word light has an I in it. So an I band, what's weird about an I band is that it spans 2 sarcomeres. So the I band is the part that's free of myosin. So the I band is light. That's how you can know that. There's an I and I band. This is free of myosin. And it's there's half an I band in 1 sarcomere. I mean, you could be like, well, wait, there's half there and there's half there, so there's a hole. It's true, but they're not adding together. So half of the I band here, half of the I band here, our next sarcomere would be there. Got it? So that what happens when we contract is our H zone is going to disappear. Our I band is going to get very small. And so that's like things that they ask in an MCAT. What would you see? A, the disappearance of the A band. B, the disappearance of the H zone. C, the shrinking of whatever. And what you would see is the disappearance of the H zone. The A band never goes anywhere. So all it does is, I mean, because this is where we're thick and thin are overlapping. So our A band is going to just kind of get darker as we get more and more overlap and we push those Z discs together. I guess I shouldn't say darker. We'll just have more overlap and then we'll get rid of this H zone altogether so that our A band comes closer and we bring our Z disc closer together and lose that I band. So actin filaments are our thin filaments. Again, they have the regulatory components that are going to move off when calcium is present. And then our thick filaments have our myosin that are pointing out in that direction. Okay, any questions so far? Yeah.
What are the rectangle things on
This is all thick filaments.
here? Okay.
So these are just thick filaments. So if I were to zoom in there, it would be where I have all of my tails. And then all my heads are poking off of it. But to draw a diagram, or to draw a sarcomere, I wouldn't have you draw every myosin head, but I would have you be aware that these are thick. So my thick filaments are anchored to my M line and they're pointing out. My thin filaments are anchored to my Z disc and they're pointing in. So that what happens when I'm contracting and I move these regulatory proteins, my actin binding site on my myosin head can reach up and grab onto my myosin head binding site, and then these things pull. And my myosin heads just start pulling and pulling, and when we pull and pull and pull until we pull our Z discs together and shorten. So that's where we're going. But we'll get there tomorrow. The last part of anatomy that I want to talk about today is the sarcoplasmic reticulum and the T tubules. Because what this is going to do, this arrangement of the plasma membrane, like creating these T tubules or transverse tubules, invaginations, poking in and around those myofilaments, is going to be what allows us to get an action potential into this muscle. And then we need to get that calcium out of that sarcoplasmic reticulum and interacting with those sarcomeres so that we can get shortening. So, any questions so far? All right. Then this is the last thing we'll talk about today, and then you can continue working on your skeleton lab, looking at your knee joint, all of that stuff. So, if you look into a muscle fiber, we can see that we have these things called triads. So again, here's my muscle fiber. I shouldn't have changed colors, but whatever. Here is my sarcolemma, and here's my T tubule, and here's my sarcoplasmic reticulum, and it's going to form this other important anatomical feature called the triad. So, if we think about what's going on here, in our sarcolemma, we have all of the voltage-gated channels that are going to give us an action potential. So our sarcolemma contains voltage-gated sodium channels and voltage-gated potassium channels. So that an action potential is going to come from our lower motor neuron and stimulate an action potential in this muscle fiber. So the action potential in the muscle fiber is going to go down the sarcolemma and it's going to come down these T tubules. So that my action potential is going deep into my muscle fiber and surrounding, like it has the potential then to interact with each of my myofibrils. Another thing that we find as part of this triad, well, it's two things. One, two, what we call terminal cisterna of the SR. And it's just these enlarged regions that flank a T-tubule. And so here's one enlarged region, here's one enlarged region, all three of them, 1, 2, 3, make a triad. So a T-tubule and its two terminal cisterns are a triad. And what that allows to happen is that we can conduct this action potential down the sarcolemma and deep into here. And then, when this action potential is in here, it's going to be able to open up these voltage-gated calcium channels here. We've got calcium floating around stored inside the sarcoplasmic reticulum. When the voltage-gated calcium channel opens, that calcium can come out and flood the sarcoplasm, and now it can move those regulatory proteins and we can start contraction. So that's kind of the last part of the anatomy that matters, is that our sarcolemma is creating these transverse tubules so that we can come in and surround our myofibrils, which is where our proteins for contraction are. So if I've got a bunch of calcium in here, I can open these voltage-gated calcium channels, calcium can flood out in here, move the troponin and tropomyosin complex, and I can start contraction. Okay? That's the anatomy, and that's all we're worried about today. Those are the structures that give rise to the function.